We're going to take a look at Gregor Mendel's experiments. He was an Austrian monk who was doing science experiments and was very interested in genetics, although they didn't call it that back then. And he was interested in how things are inherited and passed on to offspring. They didn't know about DNA and genes back then, but he figured out quite a bit without knowing all that. So here's Gregor Mendel right here. And because of everything that he figured out, he's known as the father of genetics. When Mendel first started working, what they thought about inheritance was that the genetic material from the two parents would blend together and that the genes would not remain separate. That is totally wrong. But you can see why they used to think that, because when you look at yourself and compare yourself to your parents, you kind of look like a blend between your two parents. So that's what they thought was going on, but that's actually not correct. Mendel thought there was something else going on, and he came up with this particulate hypothesis. He didn't know about genes yet. They didn't know that, so he called genes particulate. So what he said was that genes are going to be inherited from each parent, and they're going to remain separate. They don't blend together. And this is why we'll see some traits that will disappear in one generation because maybe they're recessive and they get masked or hidden by a dominant allele, but then they'll reappear in a following generation because then they get unmasked because there's no longer a dominant allele with them. Okay, Mendel chose to work with peas, which was actually a good choice because there's lots of different traits that can be studied on peas, and there's a very short generation time. You can grow the peas and get seeds very quickly and then plant those and get new plants, and there's a large number of offspring that are produced, so you get lots of pea seeds that you can plant. And then also peas are self-fertilizing. The peas and their flowers have both male and female parts, and Mendel could actually control the pollination, so he could actually control which plants that he wanted to cross. These were all of the traits that Mendel studied, so there were quite a few things that he looked at. And then just a couple more terms here. So true breeding plants are ones that are homozygous for a particular gene. So for example, if we're looking at flower color, you would have, for example, these homozygous dominant purple flower true breeding plants. And if you crossed two true breeding purple flowered plants, all of their offspring are going to have purple flowers. Or if you crossed two homozygous recessive white flowered pea plants, all of their offspring would have white flowers. So true breeding plants will just give you the same color flower offspring as what the parents had. Okay, hybridization is when you're going to cross two different true breeding varieties. So he would cross a purple flowered plant with a white flowered plant. And then all of the offspring would be hybrids or heterozygous. So they would be a big P, little p, would be their genotype, and their phenotype, what they would look like, they would all have purple flowers because the big P, the purple allele is dominant, and so they all would be purple. Okay, a few more definitions. The P generation, P stands for parental. These are going to be the true breeding parents. And then the F1 generation, this will be the offspring of the P generation. F just stands for filial, and filial just means offspring. Okay, and this will be our first generation of hybrid offspring. So the one is just the first generation. And then what he did was he would cross two of these F1 hybrids, and then he would get the F2 generation. So this would be our second generation of offspring. Okay, so this is the results of Mendel's experiments. The P generation, he would take the two true breeding purple and white flowered pea plants, cross those, and then here's the F1 generation, our hybrids, which are all heterozygous. And then we would cross two of these hybrids, and then here would be our F2 generation. And so he noticed that the F1 generation that are all purple, he discovered that, oh, they're all purple because the purple allele is dominant and the white allele has to be recessive. So he discovered that. And then he also figured out that there's not blending going on because in the F2 generation, the white allele reappeared. So it didn't mix together with the purple allele. It's still there, it just was hidden by that dominant purple allele. Okay, so Mendel came up with two laws with his theory of inheritance. He came up with the law of segregation and the law of independent assortment. And both of these laws are due to meiosis and the formation of gametes. So we're going to take a look at the law of segregation first. So if we start with our diploid germ cell in the ovary or the testes, and we're just going to be looking at one particular gene, maybe this is a height gene, and what we're going to be doing during meiosis is, remember, we're going to be making our haploid gametes. So what we're going to be doing is we need to take our diploid cell and we need to cut the number of chromosomes in half. Okay, so we're going to be dividing twice. So in meiosis 1, our first division, we're going to separate the homologous pairs of chromosomes into separate cells. Okay, and notice that we've separated these two alleles. We've got one chromosome with the big T allele and the other chromosome with the little t allele. So these end up in separate cells. Okay, and so that's going to be our law of segregation, is we're just going to segregate or separate these two alleles into separate cells. Okay, so big T ends up in one cell and little t ends up in another cell. And then our second division with meiosis, we then separate our sister chromatids, and you can see that half of our gametes will have big T and then the other half of the gametes will have little t in them. Okay, so that's just our law of segregation. We're just looking at a single trait, a single gene. We're just maybe looking at this height gene, and we're just going to separate these two alleles, big T and little t, into separate gametes. Okay. Okay, and then when the sperm fertilizes the egg, these two alleles will then be rejoined together, and now our zygote, our fertilized egg, will be diploid again. So we have our haploid sperm and egg. They only have one copy of each of these chromosomes, but now they're diploid again. They've got a homologous pair again. Okay, so how Mendel figured out this law of segregation is he did what's called a monohybrid cross. Okay, so mono just means one, and hybrid, remember, means heterozygous. Okay, so how we figured this out is in this, to get this F2 generation here, he took two of these hybrid purple flowers, and they're each heterozygous, and when they go to form the egg and the sperm, remember we're going to do our law of segregation. So we're going to put these into separate gametes. So half of the eggs will have big P, the other half of the eggs will have little p, and then for the sperm, half of the sperm have big P and half the sperm have little p. Okay, so we're going to form this, what's called a Punnett square. And so we've got, maybe we'll put the eggs up here on the top, and then these would be our sperm here. And then how we do this Punnett square is we're just going to fertilize this sperm and this egg together. And this could be one possible offspring. And then this egg and that sperm could come together, and that could be a possible offspring. And then this egg and that sperm can come together. That's another possible offspring. And then our last possibility could be this egg and that sperm, and there's our last possible offspring. Okay, and then so we have 4 different possibilities for our offspring. And if we look at our phenotypes, what these look like, you can see that there's 3 purple offspring to 1 white offspring. Okay, so that would be our phenotype ratio, 3 to 1. And then our genotype ratio, we would just look at, well, what's the genotypes of our offspring? Well, we've got one that is homozygous dominant, 2 big P's. And there's 2 that are heterozygous. And then there's 1 that's homozygous recessive. Okay, so we've got a 1 to 2 to 1 genotype ratio. Okay, so by looking at these numbers, Mendel was able to figure out this law of segregation. So he's like, oh, well, to get these ratios, that means that these 2 alleles have to be separating from each other and ending up in separate gametes. Okay, and this is just looking again at the ratios with the phenotype, what you look like. You've got 3 purple offspring to 1 white offspring. And the genotype ratio, you've got 1 homozygous dominant to 2 heterozygous offspring to 1 homozygous recessive offspring. Okay, so let's just do some practice with our Punnett squares. You always want to make a key. And this will help you out. So we're going to have our height gene. And we've got 2 alleles. A big T means you're going to be tall, and a little t means that this plant is going to be short. Okay, so let's say that we're going to cross these 2 parents. Okay, so we've got a heterozygote and then a homozygous recessive. So with our phenotypes, this one would be tall and this one would be short. Okay, so now we're going to make our Punnett square. Okay, and one parent. I know, we'll say that this is the female, the tall. Okay, we do our law of segregation, and half the eggs will have big T and the other half will have little t. Okay, and then let's say the male plant will be short, and half of the sperm will have little t and the other half will have the other little t. Okay, and then we can do our Punnett square and just fill out big T and little t go together, and then these two little t's go together, and then this big T and this little t go together, and then these two little t's go together. Okay, and then we could ask, what percent of the offspring would be tall. So the tall offspring we have 4 possible offspring, and 2 out of the 4 are going to be tall. And if we get that to our lowest common denominator, that would be half, or 0.5, or 50% would be tall. And if we ask about, which ones are going to be short offspring, it's the same answer, because we've got 2 out of the 4 that would be short. Okay, or if I asked, what's the probability that you would get an offspring that's homozygous recessive? Well, you'd have 2 out of the 4 that are homozygous recessive, or 1/2, or 0.5, or 50% that are homozygous recessive. Okay. Another example. Let's say we're going to work with some guinea pigs. And let's say we've got a hair length gene. Actually, let's do a hair color gene. And let's say big B is going to be dominant, and then I'll give you black. And let's say little b will be recessive, and that would give you, let's say, kind of a reddish orange color. Okay. All right, so let's say that we've got our 2 parents, and let's say one parent is that one, and the other parent is this one. Okay, so one parent is homozygous dominant, the other one is heterozygous, and their phenotypes, what they both look like, they're both going to be black in color. Okay, so now we're going to make our Punnett square. And let's say this is our female. So if we do our law of segregation, half the eggs will have big B, and the other half will have the other big B. And then let's say the male is the heterozygote, and so half the sperm will have big B, and the other half of the sperm will have little b. And then we do our Punnett square, and these two go together. And then those two go together. And then these two go together. And then those two go together. Okay. So the probability of getting a red offspring okay, remember to get a red offspring, it would be two little b's. They'd have to be homozygous recessive. And that'd be 0, right? You wouldn't have any of those. So there's no homozygous recessives. And the probability of getting a black offspring, well, all of them are black, right? All 4. So it is 1 or 100%, right? Okay, and the probability of getting a heterozygote offspring. Okay, well that would be 2 out of the 4, or 1/2, or 0.5 or 50%. Okay. Okay. Sometimes the phenotype of an individual doesn't always tell you what its genotype is. Okay, so if you have a white-flowered pea plant, you know its genotype. Okay, it has to be homozygous, whoops, we're using peas. It has to be homozygous recessive. You've got 2 little p's there. Okay, and a lot of times for my lowercase letters, I'll put a little line through them just so I remember that they're lowercase, because sometimes lowercase and uppercase look pretty similar for some letters. So I'll put a little line through it. Okay, what about the genotype for a purple flower? Well, it could be homozygous dominant, or it could be heterozygous. Both of those would be purple. So to figure out the genotype of a dominant phenotype, like that we've written up here, you can perform a test cross. Okay, so what we do in a test cross is you're just going to cross that dominant unknown genotype with the recessive genotype. Okay, so this purple flower could be homozygous dominant or heterozygous. You don't know yet. And you're going to cross it with the recessive phenotype or homozygous recessive. And when you do those crosses, if you get any offspring that have the recessive phenotype that are white, then you know it's a heterozygote. But if all the offspring have the dominant phenotype, like if they're all purple, then you know it had to have been the homozygous dominant was what your purple flower was. Okay, so that's a test cross. So it just helps you figure out what the dominant phenotype is. This purple flower is it homozygous dominant or is it heterozygous? To figure that out, you do a test cross.